Now let's talk about cytoplasm and briefly discuss the organelles in the cytoplasm. Cytoplasm is a thick gel-like semi-transparent fluid that is found in both plant and animal cells. And the constituent parts of cytoplasm are cytosol, cell organelles, cytoplasm nucleus, bounded by plasma membrane, contains many organelles in eukaryotic cell. Eukaryotic cells are the cells in which there is a clear nuclear membrane. Cytosol is the aqueous part of the cytoplasm. Outside all of the organelles, it also contains its own distinctive proteins. It accounts for almost 70% of total cell volume. Gelatinous cell substance consisting mainly of cytoskeleton filaments, which give strength to the cell. Mostly organic molecules, salt and water. Chemically, the cytoplasmic matrix is composed of many chemical elements in the form of atoms, ions, and molecules. The organelles found in the cytoplasm are mitochondria, endoplasmic reticulum, lysosomes, Golgi apparatus, peroxisomes, vacuole. Each organelle is bound by a lipid membrane and has specific functions. Let's talk about mitochondria, which was first observed by Colliker in 1850 as granular structures in the striated muscles. Mitochondria are called the powerhouse of the cell. There is the inner membrane making the cristae, and the matrix is there inside the inner membrane, and then outer membrane. It covers the whole mitochondria. Its length is around 5 to 12 micrometer, diameter is 0.5 to 1 micrometer, filamentous or globular in shape, and outer membrane, inner membrane are the structures or components of this mitochondria. There is intermediate space between outer and inner membrane. Cristae is there. These are the foldings of the mitochondria. Let's do it clearly. Okay. Matrix, the space enclosed by inner membrane is called the matrix. And then comes the membranes of all of the inner and outer membranes are made up of phospholipids and proteins. Outermost membrane, it contains large number of integral membrane proteins called porins. Right? Multiple proteins called porins. These porins form channels that allow molecules of 5,000 daltons or less to pass. Studied with enzymes concerned with biological oxidation. This outer membrane, it is having multiple enzymes. They help biological oxidation. The processes of respiratory process is all happening inside the mitochondria. The interior, the matrix of the mitochondria contain enzyme concerned with citric acid cycle and respiratory chain oxidation, or ETC. Major metabolic pathways involved in oxidation of carbohydrates, lipids, and amino acids are part of special biosynthetic pathway involving urea and heme synthesis, or are located inside the mitochondria. Inner membrane is the place it contains ATPs and other enzymes concerned with synthesis and metabolism of ATP. It contains enzymes for electron transport chain, or it contains proteins for electron transport chain as well. The ultimate purpose of these mechanisms is oxidative phosphorylation and synthesis of ATP. Mitochondria has some protein synthesized by mitochondrial DNA. Functions are they generate power for the cells. They are important to maintain proper concentration of calcium ions within the various compartments of the cell. Energy transduction through respiration they are responsible to regulate the energy of the cell. Next comes the ER, or endoplasmic reticulum. These are network of tubular and flat vesicular structures in the cytoplasm. An extensive network of closed flattened membrane. These are the flattened membranes, closed, very near ones. These are not cylindrical ones. An extensive closed membrane bounded sacs called cristae are there, and the space inside the tubes is filled with endoplasmic matrix. There are two types of ER, smooth and rough endoplasmic reticulums. In smooth endoplasmic reticulums, there is no ribosomes on the surface, but the rough endoplasmic reticulum, they have ribosomes which help for the synthesis of protein. And SCRS mainly present in lipid-forming cells such as adipocytes, intestinal cells of testes, glycogen-storing cells of liver, adrenal cortex cells, muscle cells, leukocytes, etc. However, ER are mostly present in protein-forming cells such as pancreatic acinar cells, globe antibody-producing cells, nacelles, granules of nerve cells, etc. The functions are to synthesize proteins, protein segregation, unsaturation of fatty acids. Fatty acid cycle also happen in ERs. Muscle contraction, ER is commonly known as sacroplasmic reticulum in muscle fibers. Then comes Golgi bodies. Golgi bodies is a collection of membrane enclosed sacs. These are Golgi intermediate compartment, Golgi network. These are all called Golgi states. These are plasma membrane secretions. It's a collection of membrane enclosed sacs composed of 4 or more stakes layer of thin, flat enclosed vessels lying near the outside of the nucleus. They consist of these structures, they lie near to the nucleus, let's say, and they consist of multiple discrete compartments. These type of compartments, and here, and the cis Golgi network consists of 4 functionally distinct regions. 4 regions: cis Golgi network, Golgi stack, which is divided in Golgi stack, this one, medial and trans subcompartments, and the trans Golgi network.